Hi everyone. Welcome. Today we're going to be talking about the part two of the circulatory system. I'm just going to give it a couple more minutes. Today's session should be short anyway, since we're just finishing up. Okay, I'm going to get started. If you guys can hear me, can you just say hi in the chat? Okay, thanks, Sarah. So today we're just going to be finishing up the part two of the circulatory system. If you guys didn't get to go to that session, we always send out slides and recordings. So if you didn't get it, you can email us and ask for it. Okay, so I'll just get started. So the kind of the stuff that we have left to discuss is some of the more detailed parts. So rather than the big picture, like higher yield stuff, this is more so the details. So first part of it is just looking at what makes up blood. And like the two big divisions at first are plasma, so like the liquid part of blood, and then the cellular components. So within plasma, then there's different types of nutrients and salts, gases, so oxygen, CO2, other gases, hormones, and blood proteins. So, and then the cellular components are different types of cells. So erythrocytes are red blood cells. You have leukocytes and then also platelets. So to go a little bit more into the cellular component of blood, these are the different types of the red blood cells, and their function is primarily oxygen transport, and they do this via hemoglobin. And so kind of like the important characteristics to know about red blood cells is that they're biconcave. And this just refers to the shape of them. And they also don't have any membrane-bound organelles. And they're constantly renewed and recycled in the body, so a single red blood cells doesn't undergo mitotic division. It just dies after its life cycle, which is about 120 days. And there's different ways of measuring hemoglobin when we talk about red blood cells. So you can either look at hemoglobin quantity, which is looking at grams per deciliter of blood. This actually involves taking an arterial blood sample, so it can be an invasive procedure. So usually the other measurement is used, which is hematocrit, and that is the measurement of what percentage of the body's hemoglobin is oxygenated and carrying oxygen, and what percent is deoxygenated. Okay, so that's that for the red blood cells. To move on to leukocytes, which are white blood cells. Their primary function is in the immune system, so to cause inflammation and fight off infections. And there's various different types of leukocytes that are listed on the slide. I'm not going to go into too much on the details here because this is a big part of the discussion on the chapter on the immune system. We have a topic session coming up next week on the immune system as well. So pay attention to that if you want to learn more about this. And then the last types are platelets, which are also known as thrombocytes, and their primary function is to clot blood when there's damage to the blood vessels. And even though these are called blood cells, or they're categorized under blood cells, they're actually not fully functioning cells, they're cell fragments. I've seen a couple questions come up on that, so keep that in mind. And just this note I have over here, all blood cells originate in the bone marrow through hematopoiesis. So this is just a process that generates all the cells in the body. Any questions on that? All right. So next thing to discuss when we talk about blood are blood types. And blood is categorized by the antigens that we have in our blood. So antigens are proteins that are on the cellular surface. So there's two different types of antigens. There's the ABO antigens, and then there's the Rh factor. In ABO antigens, you have three types, A, B, and O. A is also represented as an allele with the capital I superscript A. B is represented with the capital I superscript B. And then the O is represented with the lowercase I. Sometimes you'll also just see A, B, and O for alleles, but this notation is way more common. And then in the Rh factor, you just have two types, positive and negative. So ABO antigens exhibit both codominant and regular Mendelian dominance. So A and B are codominant, while O is recessive to both A and B. So that's how you end up with AB, because when you have one allele of A and one allele of B, you have an AB blood type because they're codominant. However, because the O allele is recessive to both A and B, as long as you have one A allele and then you have an O allele, you will be an A blood type. If you have one B allele and then one O allele, you'll be a B blood type. And obviously, if you have two alleles that are the same, that will be A and B respectively. And the only way to have O is to have two little I O alleles. And then in the Rh factor, the inheritance pattern is more straightforward. Negative factor is recessive to the positive factor. So if you have one positive and one negative, you'll be presenting as a positive blood group. There's a question in the chat, aren't antigens foreign substances like bacteria, etc.? So antigens are just cell surface proteins, and you're probably most familiar with antigens on like foreign substances like bacteria, and that's what causes like an immune response because our body recognizes these foreign antigens. But our bodies also, or our cells in our body also have antigens that our body recognizes as our self antigens. Does that help? Okay. Let me see. Okay. So if you want to look at this chart on the right hand side, or here, this one's the same one, so I'll move on to this slide. So this is a representation of the blood groups with the pictures of the antigens kind of drawn out. So the A, think of these little green triangles as the A antigen, this orange one as a B, and then O is actually represented by no antigens. So the O allele doesn't code for any antigens. So on a group A blood type, on a red blood cells in a group A blood type, you will have just these green A antigens. In group B blood type, your red blood cells will have these orange antigens on their cell surface. AB will have both, and then O will have none. So you have antibodies. To move on to this row, your body produces antibodies against antigens that your own blood doesn't have. So if you're blood group A, your body recognizes A antigen as your self antigen, so it doesn't produce antibodies against A. But you don't have your B antigen, so your body does produce antibodies against B antigens. So that's the opposite for blood group B. Your body recognizes B antigens as your self antigens, but not A antigens. So it produces antibodies against A antigens. So next in the AB group, you have both A and B antigens. So your body recognizes both of those as self antigens and doesn't produce antibodies against either. Lastly, if you're an O blood group, your body doesn't produce either the A or the B, your red blood cells don't have either the A or the B antigen. So your body produces antigens against both A and B antigens. And this is important because this all comes into when you're looking at blood transfusions, which type of blood you can accept blood from. So because type O doesn't have any antigens on its surface, no matter who you give the blood to, if they're A, B, or AB, there's no antigens on the cell surface of an O blood group red blood cells for the body to react against. So that is called the universal donor. And this chart only is looking at ABO antigens, but if we were to also look at the Rh factor and consider positive and negative blood type, the universal donor is O negative. So O positive has the Rh factor on its cell surface, so that's why you can't give it to a negative type blood, because that negative type bloods have antibodies against positive Rh factor. On the other hand, the universal recipient is type AB. So you see that this blood type has both antigens on its cell surface, which means that it doesn't produce antibodies against either of those antigens. Therefore, you can give it any blood type and it won't produce antibodies against it. And to go into the Rh factor, this is AB positive, that's the universal recipient, because if you gave a AB negative person a positive type of blood group, that transfused blood will have an Rh factor antigen on the surface of the red blood cells. So the negative group would has antibodies against this. Okay, so to explain the Rh factor again, let me go back here. So the Rh factor is another type of antigen. You have ABO antigens on your blood, but you also have Rh factors. So it's just another type of self-surface protein. So how you have ABO antigens, in the ABO antigen groups, there's 3 types of alleles, A, B, and O. A codes for A antigen, B codes for B antigen, and O doesn't code for any antigen. Similarly, on the Rh factor side, you have 2 alleles, positive allele and a negative allele. So positive codes for the Rh antigen, and a negative allele means that you don't have Rh factor antigen on your red blood cells. Does that help both AJ and Jane? And you can unmute yourself if you want to ask a further question as well. Okay. Does anyone else have any other questions? Okay. So this chart is kind of like another similar chart. So I'll just let you look at it. It kind of just more clearly lists out what blood types can donate to which other blood types and take blood from. Okay, so the next topic that we have is discussing what blood pressure actually is. So blood pressure is simply the pressure exerted by blood on the wall of your blood vessels. So the reason why it's important to have a high enough blood pressure is that the circulatory system's entire role is basically to move oxygenated blood throughout the body. So you need blood pressure that's high enough to move that blood forward and to keep it going throughout the body. However, hypertension is what is a name for having too high of a blood pressure, and this is a problem because too high of pressure can damage blood vessels and organs as well. And just like a note on the unit, it's force per area, it's the same unit, force per unit area, it's the same unit as any pressure measurement in physics. And it's expressed as a ratio of the systolic to diastolic pressures. So we talked about what systolic and diastolic means in the last session. And the instrument is this sphygmomanometer. And the important thing to note is that pressure gradually drops from arterial to venous circulation. And so I have a picture on this slide. So you can see that when you start off at the aorta, and if you guys remember, this is the first major blood vessels that carries out oxygenated blood from the left ventricle to the body. So here's when your blood pressure is highest. So this is an example pressure. So say 100 millimeters of mercury. So this is a unit of pressure. And as you proceed through your arteries, you're kind of maintaining that pressure pretty much. But as you enter these really small blood vessels, arterioles, capillaries, and venules, your pressure drops a lot, and then the pressure is lowest in going through the veins, it's lowest at the vena cava. So why this drop in blood pressure occurs is because these tiny blood vessels are so small in diameter that they put up a resistance against the blood pressure. Important to note that the largest drop actually occurs at the arterioles and not capillaries. And then this is the direction of blood flow going from oxygenated blood. And then this would, like, your capillaries are where your target organs or part of the body would sit. And then so the venules would take away deoxygenated blood. As to why it's the arterioles, I think it's because it's the first kind of move from going from bigger blood vessels to smaller blood vessels. So because the arteries, so when you're entering from the arteries, the transition to the arterioles is moving from a really big diameter blood vessels to smaller ones. So that initial resistance drops this blood pressure a lot. When you're going from arterioles to capillaries, arterioles are pretty similar in diameter, so that change in blood pressure is a lot less significant. So I think it has to do with the order of which the arterioles are in comparison to the capillaries. But yeah, that's a common confusion. You're welcome. So next thing to discuss is how blood pressure is regulated in our body. So this is a huge deal, and just like clinical management, a lot of people have hypertension, and it's like a big deal, like a chronic disease that's increasing in prevalence more and more. So it's kind of, there's two different types of neurons that regulate blood pressure. First is the baroreceptors. So baro means pressure. These sense just blood pressure directly in the walls of your blood vessels. So once these receptors sense that drop in blood pressure, the sympathetic nervous system kicks in and causes vasoconstriction. So this is constriction of the blood vessels, and this increases the blood pressure. There's also chemoreceptors, which instead of sensing pressure, they sense osmolarity, and they cause the release of various hormones that can either increase or decrease blood pressure. And there's a lot more detail on these hormones in the renal system chapter that we also did a topic session on. So I'll direct you to those so I don't spend too much time on these. ADH and aldosterone both increase blood pressure, and ANP decreases blood pressure. But instead of just memorizing that, knowing those mechanisms is also important, and it's frequently tested. So I encourage you to take a deeper look at that if you don't know the mechanisms of how they increase blood pressure. Okay. So this kind of goes back to when we were talking about the red blood cells, and their function is to carry oxygen. So how does gas exchange actually work? Is that a gas exchange of oxygen in particular is that there is some oxygen that diffuses into the blood and is just carried that way. But this is a very small amount compared to the oxygen that's actually carried by hemoglobin. And there's several ways to measure the amount of oxygen in your blood. There's the partial pressure of oxygen, but then there's also oxygen saturation. Both of these are I have listed here. So partial pressure of oxygen is just the comparative pressure of oxygen to everything else in the blood, so all the other gases in the blood. And this is basically like measuring oxygen concentration. Oxygen saturation, on the other hand, is a percentage of hemoglobin carrying oxygen. Okay. And then how oxygen is actually carried by hemoglobin is that hemoglobin has these heme groups, which is this conjugated ring containing iron in the middle. So that heme group iron can bind oxygen directly, and that's how hemoglobin is able to carry oxygen. So when you bind and release oxygen, this is actually an oxidation and reduction reaction. So the iron in the heme ring has two oxidation states. There's the 2+ oxidation state and the 3+ oxidation state. So the 3+ oxidation state is when the hemoglobin has actually bound to oxygen, and then the 2+ is when it's not. So something to keep in mind is that hemoglobin can only bind oxygen when the iron is in the 2+ oxidation state. And I'll give you guys a minute here if you have any questions. And then, sorry, Mythili, I didn't see that earlier. Yes, ANP is produced by heart cells. Does anyone else have any questions? What is attached to oxygen? This looks like it's another, oh, this is just a hydrogen bond to a neighboring heme molecule. So it's not actually attached to it. These are just intramolecular interactions. Is that what you're talking about, Crystal? This one? Okay, yeah. Okay, and then I just realized that I misspoke earlier about something, so I'll go back to that slide just for a quick second. So hematocrit, I said it incorrectly earlier, so please note this down if you wrote down the meaning. So hematocrit is actually the proportion of red blood cells in your blood and not how much hemoglobin is carrying oxygen. So it's the proportion of blood that's carrying red blood cells to the percentage that's not. Sorry about that. So I'll go back to the slide. So let me take Mythili's questions first, and then I'll go back to AJ. So when oxygen binds, the planarity of the heme does change. And there's another slide coming on cooperative binding. Is that what you're talking about? And I'm sorry if I'm saying your name wrong too. Yeah, it's a relatable concern. Sorry. And then AJ, what do you mean by Fe3+ being stronger? I don't think that's really relevant to the discussion. I'm still not understanding what you quite mean. Sorry.
Are you talking about my question?
Yeah.
Do we need to kind of memorize the structure of the hemoglobin and the iron?
No. Like this heme group thing?
Yes.
No. Just know that the two different oxidation states, and that when the iron is in the 2+ state, it's unbound to oxygen, and then when it's in the 3+ state, it is bound to oxygen.
I see. Thank you.
And then to keep talking about cooperative binding, to keep talking about oxygen and hemoglobin, an important concept is also what is called cooperative binding. So each hemoglobin has 4 subunits of four subunits. So this is a peptide chain, peptide chain, peptide chain, peptide chain, and this all makes up one hemoglobin protein. So each of these 4 subunits have a heme group. So each heme group can carry an oxygen molecule. So once the first oxygen molecule binds the first subunit, what happens is that the entire protein goes through a conformational shift. This is a shift from like what's called a tense state to a relaxed state. And as we were talking about earlier, there's this shift that happens in the molecule structure that allows for easier binding of the second, third, and fourth oxygen molecules to the second, third, and fourth subunits. So basically, the first oxygen binding makes it easier for the second, third, and fourth oxygen to bind a single hemoglobin molecule. So similarly, when you have a situation where all four heme groups are bound to oxygen, and then now you need to now that hemoglobin needs to let go of that oxygen, it's easier for the oxygen to leave the subunits once one of this oxygen has already left. So when the first oxygen leaves, there's again a conformational change that occurs that makes it easier for the latter oxygen molecules to leave. And we also did a topic session on enzyme kinetics, which talks about this in a little bit more detail if you want to check that out. Okay. And then this is a kind of like an important graph to be able to read. So oxygen has different affinity for carrying oxygen based on the partial pressure of oxygen in the blood. So when you're at the lungs, that's when you have the highest partial pressure of oxygen. So at that point, hemoglobin has a really high affinity for oxygen, which means it'll be really saturated with oxygen. On the other hand, when you're at tissues at rest, so these are like tissues on other body organs, your partial pressure of oxygen is much lower than what it is in the lungs. So your hemoglobin lets go of that oxygen to supply the tissue with oxygen. So that's why its oxygen saturation is lower when the partial pressure of oxygen in the tissues is lower. So that's like when you're at rest and not doing anything strenuous, but when you exercise, your partial pressure of oxygen drops even more. So at that point, the hemoglobin is able to recognize that and dissociates even more oxygen. So that's when its oxygen saturation drops to its lowest point. Is everything okay with that? Okay, thanks, Brian. Thanks. All right. So we talked about gas exchange of oxygen. Another important one is CO2, carbon dioxide. So similar to oxygen, is some CO2 that's dissolved in the blood, but a minimal amount. And then some CO2 is also carried by hemoglobin, but hemoglobin has such a greater affinity for oxygen that this amount is also minimal. So how CO2 is actually exchanged is that it's done in the form of carbonate or carbonic acid. And this is the majority of the way, this is how the majority of carbon dioxide exists in blood. So this is kind of the reversible reaction that generates carbonic acid and then the carbonate ion. Sorry, bicarbonate ion, not carbonate. So you have this enzyme called carbonic anhydrase that catalyzes this reaction. So what you can notice is that concentration of carbon dioxide changes the concentration of protons in the blood, and proton concentration affects pH. So all this to say, CO2 concentration in the blood is able to change blood pH. So for example, when your carbon dioxide concentration of the blood increases, you produce more carbonic acid, so therefore more protons. So when your proton concentration increases, your pH drops. Your blood becomes more acidic. On the other hand, when CO2 decreases, proton concentration decreases, pH decreases, and your blood becomes more alkaline. So pH is a really important factor to maintain steady in the blood. So that's why carbon dioxide needs to maintain at an important, at a very narrow level in the blood also. One of the other reasons. Okay. So we're talking about how carbon dioxide concentration is related to proton concentration. And one of the things that's important is because it deals with blood pH, but another important factor is that protons actually can bind hemoglobin, therefore reducing hemoglobin's affinity for oxygen. So when there's increased concentration of protons, there's reduced hemoglobin affinity for oxygen, which means that hemoglobin dissociates more oxygen, therefore unloading more oxygen at a particular spot in the body, such as like tissues of a particular organ. And this is actually a beneficial tool for our bodies to have. So the triggers that are involved in increasing proton concentration occur when your tissues have an increased demand for oxygen. So that trigger allows hemoglobin to unload more oxygen when it is bound to these protons. So we talked about how increased carbon dioxide concentration increases proton concentration. So these triggers include increased CO2 concentration, which increases H plus concentration, which is the same thing as decreased pH. And then increased temperature also is another trigger. So all of these occur when your body or certain tissues in your body need more oxygen. So the Bohr effect refers to the fact that when your body is under these conditions, hemoglobin will have a lower affinity for oxygen. This means that under these conditions, hemoglobin will release more oxygen to fulfill that body's need for oxygen. And on a graph like this, you'll see that in the form of a leftward shift of the hemoglobin dissociation curve. Sorry, a rightward shift of the hemoglobin dissociation curve. So here's your hemoglobin curve under normal conditions, and then the rightward shift. So how to think about this is that pick a particular number. Let's go with 40. So when you're at 40 millimeters of mercury for your partial pressure of oxygen, under normal conditions, your hemoglobin would be saturated about 80% with oxygen. When you have these conditions such as increased CO2 concentration or increased proton concentration or increased temperature, you get this rightward shift of the hemoglobin oxygen dissociation curve. And so under those conditions, even though it's the same partial pressure of oxygen, your hemoglobin will only be saturated 40% with oxygen. So that's a huge shift regardless of the same partial pressure. Under normal conditions, it'd be 80% saturated, but hemoglobin's only 40% saturated. So that's how to think about these. Think about the Bohr effect. In certain conditions, there's also a leftward shift, which is increased affinity of hemoglobin for oxygen. So the conditions for this are basically the opposite of any of these conditions. So decreased CO2, decreased H plus concentration, increased pH, decreased temperature. And another one that might come up is this decreased 2,3-BPG concentration. And this product is, its compound is a side product of glycolysis in red blood cells. So it comes up in the metabolism chapter a little bit, but you can just know that for the purpose of the circulatory system, decreased concentration of 2,3-BPG is one of the contributors to the left shift of the hemoglobin dissociation curve. Okay, and another thing, when you look at these hemoglobin oxygen dissociation curve, is to look at fetal hemoglobin. So fetal hemoglobin has higher affinity for oxygen than adult hemoglobin. So again, if we look at like the 40 blood pressure, 40 millimeters of mercury, partial pressure of oxygen. This is, the red is your adult hemoglobin and the blue is the fetal hemoglobin. The fetal hemoglobin has a higher concentration, higher percent saturation of oxygen at the same partial pressure of oxygen. So this is represented on a left shift curve. And another way to think about these left shift versus right shift curve is that left shift curve mean that under those conditions, hemoglobin will hold on to oxygen tighter at the same concentration with lower concentrations. And a rightward shift means that hemoglobin will let go of oxygen more easily. Any questions on this? Another function besides gas exchange of the circulatory system is solute exchange, and this can be anything from nutrients, waste, and hormones. So most of this is pretty straightforward. It's just directed through concentration gradients. So just that in your blood, there's a higher or lower concentration of the nutrients than at your targeted tissue. So that concentration gradient just drives the movement of these into or out of the blood. And there's kind of like different pathways for different substances. So carbs and amino acids, when you consume them and you digest them, they are absorbed into the capillaries of the small intestine, and that's how it enters your bloodstream. So fats, when you consume them, are instead absorbed into lacteals, which is part of the lymphatic system, also in the smallest intestine. So it's slightly different path. So it doesn't enter blood vessels right away. It goes into lymphatic vessels. And then waste enter the bloodstream directly, but then travel to the kidney where they are filtered and then gotten rid of. Hormones are a little bit different. They enter circulation in whatever organ that they're produced in, and then to get to their target tissues, they either approach cell surface receptors or diffuse directly. So most of this is straightforward. It's driven by concentration gradients, either enter blood vessels or lymphatic vessels. And then most of this solute exchange is done by diffusion, except in certain cases of the hormones. All right, another important function of the circulatory system is balancing fluid. And there's two different types of fluids. These are, sorry, not two different types of fluid, two different types of pressure gradients that drive movement of fluid in and out of the bloodstream. So hydrostatic pressure is just the pressure that blood exerts against blood vessels. And then on the other hand, osmotic pressure is this pressure generated by solutes. And it's called a sucking pressure because it's supposed to suck water into the bloodstream. And the hydrostatic pressure is the opposite because it pushes fluid out of the bloodstream. And then once it pushes fluid out of the bloodstream, this goes into the interstitium. So this is a space outside of the blood vessels and then also outside of the tissues. And then once it's in this interstitium, it gets into the tissues of the body. So on the arterial end of the capillary bed, hydrostatic pressure is greater than oncotic pressure, so there's a net efflux of water, so water leaves circulation. But then as water leaves the circulation, as you travel through that capillary bed, your hydrostatic pressure decreases to the point that when you reach the venule end, hydrostatic pressure is lower than that of osmotic pressure. So at that end, there's a net influx of water back into circulation. So here's kind of like an image to illustrate that. So you can see that, sorry, this is a little bit backwards. So look, start from here. This is the arterial end. Oncotic pressure is staying the same because the amount of stuff dissolved in the blood, the amount of protein dissolved in the capillary bed is constant. Your hydrostatic pressure is higher on the arterial end because at this point, you're going to push water out of the capillary bed. And so as you keep pushing water out, hydrostatic pressure decreases, and it decreases to the point that once you reach the venule end, the hydrostatic pressure becomes lower than the oncotic pressure. And then oncotic pressure is kind of used interchangeably with osmotic pressure. So oncotic refers more specifically to pressure caused by blood proteins. And this is makes up the vast majority of osmotic pressure, whereas osmotic refers to everything that's dissolved in the blood. So the balance of these hydrostatic and osmotic pressure are called Starling forces. So it's just a name for the net pressure that is either driving water in or out of the bloodstream. So edema is this condition that occurs when fluid is improperly balanced in the circulatory system, and you end up with excess fluid out of the interstitium, so out of the blood vessels into the space between the tissues and the blood vessels. So basically, the takeaway from this is that movement of solutes, but and then in this case, fluid it occurs at the capillary level. So the capillary is really the interface between where things are getting exchanged in and out of the circulatory system. So this exchange is basically governed by pressure differentials. So the difference between hydrostatic pressure and the oncotic or osmotic pressure. And then the way to think about hydrostatic pressure, it's the same thing as blood pressure. Osmotic pressure is a little bit different because it depends instead on the number of particles or the concentration of particles dissolved in blood. So if there's a part of the body that has a higher concentration of cellular proteins, or then a different part of the body that has lower concentration of dissolved proteins, then that part of the body, that tissue of the body that contains the higher concentration of proteins will have the higher osmotic pressure. Is that okay? I think we're getting close to the end. All right, so I think this is the last topic. Sorry, 1 second. Sorry about that. So the last topic is coagulation, and this is the last function of the circulatory system that we're going to talk about. So earlier we talked about platelets and how their main function is to form a clot. And you need to form clots when there's a damage to a blood vessels, and this is to minimize blood loss out of that vessel. So how does the process of clot formation actually start? Is that it involves these proteins called coagulation factors, and then the platelets are also involved in starting that. So the process goes once that damage is detected, platelets are kind of recruited to the part of the extracellular matrix. that becomes exposed. And when we're talking about this extracellular matrix, that's in reference to these endothelial cells, which line the blood cells. So once those platelets are recruited and they attach to that extracellular side of these endothelial cells, this protein called integrin is activated. And what they do is they attach to circulating proteins. For example, this protein called fibrinogen, which recruits even more platelets. And then the last step is that these cells and proteins kind of form a network of cells and fibers that form what you can kind of think of as like a plug to that damaged part of the vessel. And eventually, once that vessel is repaired and that hole is naturally like fixed by the generation of new endothelial cells, that clot is broken down by this protein called plasmin. I don't particularly know. Do you want to say something? Do you know anything about calcium and blood clotting? Because I haven't read anything about it, at least not for the MCAT.
No, I just remember doing a passage and I got it wrong when it was saying what is the, like which one of these things uses calcium, and of course it was saying like muscle contraction and neurons, and then it also said blood clot formation. So I don't remember what it is, but that's why I was mentioning it. Maybe you know.
I do not, and I don't remember that ever coming up, but I will look that up for you guys and include that when I send out the recap email tomorrow. Thank you for bringing that up. Okay, so let's talk a little bit more about what these coagulation factors are. So as I mentioned, they're just proteins involved in the formation of blood clots. Most of these are secreted by, I don't know what this says, liver tissue factor, which is a protein that is exposed when the blood vessel is damaged and then exposes the connective tissue of it. Sorry. And I know there's, you guys probably had like that whole big coagulation classes and cascade in your classes, but really for the sake of the MCAT, you don't need to know all of those steps, as long as you know that the endpoint is the activation of this protein, prothromboprotein, to its active form, thrombin, and then that is catalyzed by this protein called thromboplastin. And then once that is done, thrombin converts fibrinogen to fibrin, and then fibrin is what forms those small fibers that form that stable clot over the damaged area. So there is a little bit of memorization here, but it's at least a lot less than what you would have to know for like class or something. Just know thrombin, prothrombin, and then fibrin, fibrinogen. I don't even think you have to really know thromboplastin. So after all that, to summarize the different functions of the cardiovascular system, one is transport and exchange of different substances. So we talked about gases, carbon dioxide, oxygen, solutes, so amino acids, fats, carbs, hormones, and then nutrients, so and then waste. And then so there's some of a role in immunity. We didn't get too much into that, but just the fact that your blood has these white blood cells that are heavily involved in the immune system is where that connects. Thermoregulation, again, I don't think we talk too much about it today, but I think we mentioned it last time, is that the dilation and constriction of capillaries and other blood vessels is able to regulate body temperature to a small extent. And then coagulation we just talked about, and then maintaining blood pressure is another important function to avoid disease. So don't be intimidated by this, but this is just a checklist that comes off of like the AMC's like list of topics. So as you're studying it, make sure that you have checked off knowing all these topics. We covered everything that's on this list in this topic session and the previous topic session combined, so you should be good to go. Does anyone have any questions? All right, so final reminders as always, if you're able to donate to our PayPal, because we are just a group of volunteers and we have some website costs and stuff, please do so, and then also join us on Facebook if you're looking for study partners. And then our email as always is mcat101questions@gmail.com. And yeah, I'll stick around if anyone has any questions. Thank you guys for coming, and email us at any time if you have any questions. You're welcome. Have a good night, everyone. And I'm sorry, I didn't realize this was going to take the whole hour, but good night. Stop sharing my screen. Stop recording.